My name is Elisa De Franco. I was born in the northwest of Italy in a small town near Turin. I clearly remember in 1999, it was all over the news that the first human genome had been sequenced, and I was like 12 at the time. And I remember like the hype and the idea that finally we could read the whole of the genome and we could find the cause of all the rare diseases. And that idea really appealed to me. So I studied biotechnologies and I worked in a genetics lab for 2 years. And then after that, I wanted to do a PhD abroad. So that's when I moved to the UK to study the genetics of neonatal diabetes. I didn't know much about Exeter at the time, but I then since realized it was the perfect place to do any kind of research into the genetics of rare forms of diabetes. So Exeter, since the 90s, have managed to put together this amazing collection of samples from all over the world, from people with these very rare types of diabetes. Really before 2000, it was known that there were some babies that were developing diabetes extremely early, so in the first 6 months of life. Before we knew about the genetic causes, they would be treated exactly the same as children with type 1 diabetes, so they would have to have insulin injections. But this was very problematic because these children were so small, but also because we now know that insulin injections for many of these children is not the optimal treatment. So it was really, really stressful and really challenging for the families. What really changed everything was when in 2004 in Exeter, the group with Andrew Hattersley, Sean Ellard and Anna Gloin found that genetic changes in a gene that is important for the potassium channels in the beta cells that secrete insulin, was a very common cause of neonatal diabetes. And it was also found that there was a drug that could be used to treat these children that were sulfonylureas. There was a drug that was already being used in type 2 diabetes, and a treatment that was much, much better for these children and completely changed their lives and the lives of their families. In Exeter, we have been working on identifying the genetic causes of neonatal diabetes for many years, and we have the biggest collection of samples from babies with neonatal diabetes in the world. Recently, we have been using more and more powerful genetic techniques like genome sequencing to identify novel genes. And this has really empowered us to go and find some genetic causes, some genes that are extremely important for the human pancreas and beta cells, but nobody before had ever thought they had anything to do with diabetes. So finding these genes is also giving us very important and completely unexpected insights into type 1 and type 2 diabetes that are much more common than monogenic forms of diabetes. Now we know 38 different genetic causes of neonatal diabetes, and we know that there are quite a few of them. Babies can have changed to a better treatment. A lot of them can be treated with tablets instead of having insulin injections. In other cases, maybe the tablets are not an option, but knowing that specific genetic diagnosis can mean that they can access life-saving treatment, like, for example, bone marrow transplantation in children with IPEX syndrome. Me and the rest of the team have been really lucky that we had the opportunity of meeting the families. So in Exeter, we have organized twice some neonatal diabetes family days, where we had the families and the children coming here. And that was a great opportunity for them to meet us, the researchers, but also to meet each other. It is such a huge honor to be recognized by the European Association for the Study of Diabetes. This is a great recognition for the work done by the University of Exeter and the Royal Devon Hospital in understanding the genetics of diabetes. I feel incredibly lucky. The research I do has such a direct impact on people living with a rare condition. And whenever me or somebody else in the team identify the one genetic change that is the cause of the disease, it's always, the first thought is always that we can't wait to be able to tell the clinician that sometimes after years of searching, we finally found the answer, and that could be shared with the family. That's always our first thought whenever we find the cause.